All right, welcome to our third video for chapter 17, going over mitosis, meiosis, and cellular division processes. In the previous video, we went over mitosis in detail and talked about the different phases and how a parent cell can divide and end up with two identical daughter cells. Now, for this video, we're going to talk about a different process called meiosis, where this process only happens in the gametic cells, the gametes, and it ends up with a diploid parent cell dividing into four haploid daughter cells. And we'll talk exactly how that process happens throughout this video. And so as we said in the second video, all cells in the human body, except for gametic cells, divide by mitosis. So your sperm and your eggs are formed through the process of meiosis. Okay, and all those body cells will have 46 chromosomes and are diploid, except for your sperm and your eggs. These cells are haploid, so they are one N. They only have one copy of each chromosome. Okay, but each chromosome is one of the 23 pairs, right? So they will have 23 chromosomes, but they'll only have one version of the 23, because remember, we have two versions of the 23 chromosomes, leading to 46 total, so 23 pairs. But in our sperm and egg, instead of having a pair for each chromosome, we only have a single one. Now, the pairs in our cells are called homologous chromosomes, right? So remember when we said, if we have chromosome 1, right, and it's replicated, you have a maternal copy from your mom and a paternal copy from your dad. And let's say these are for number 1, chromosome number 1. Now, what chromosome number 1 means is that if you zoomed into the chromosome, right? So let's say we zoomed into the chromosome here. And if you looked at and colored kind of the DNA and looked at the banding patterns, you would notice that the bands would be similar, right? So I'm just going to kind of do different widths, right? And basically, each of these sections represents a gene. And with homologous chromosomes, they have and carry the same genes. That's super bad. Let me try again. Okay. So that means that this one, they will have the same banding pattern, right? And so these are homologous. So they're both for number 1. And so what this means is, like, let's say that this small band here is the gene for eye color. Now, because they are the same, they're homologous chromosomes, they have the same genes, right? So like this is eye color, maybe this is hair color, and maybe the bottom one is like height, right? These are all random. This is not exactly how it happens, but this is just like an example, right? Now, that means that the paternal copy and the maternal copy, this will always code for that specific trait. But now let's say for the dad, you have the blue eye, and for the mom, maybe you have green eye trait, right? And so you have a different version of those genes, but they're the same gene. They're the eye color gene, right? So that means that they are homologous. So they're coding for the same things, okay? And so we have 23 pairs of chromosomes, so 23 homologous chromosomes. 22 of those are autosomes. One pair of those are sex chromosomes, so X and Y's, right? So it's double X in females and XY in males, right? So this is basically the chromosome that determines gender. And in meiosis, the cell division process is generating the gametes that are needed for reproduction, for sexual reproduction. Okay. And so your gametes again are sperms and eggs. And like we mentioned, those are haploid cells. So they only have one set of those 23 chromosomes. And so again, they're going from a 2N parent cell that's diploid to daughter cells that are 1N, that are haploid. So going from 2 copies of a chromosome to 1 copy. And this is a cell division process that either occurs in the ovaries or the testes, depending on your gender and reproductive system. Now, meiosis, like I mentioned at the beginning of the video, it includes 2 cell division processes. So it actually divides, the cell divides 2 times. So if you have one parent cell, it will divide 1 time into 2 cells, and then those cells will divide, and you end up with 4 daughter cells. Okay, so 1 cell turns into 4. And it happens through 2 phases that is known as meiosis 1 and meiosis 2. Meiosis 1 is very different from mitosis, but meiosis 2 is somewhat similar to mitosis. Okay, and how kind of the sister chromatids divide in that cell division process. And so again, meiosis reduces the number of chromosomes by half. So daughter cells are haploid. And by this first division phase, the cells are haploid. So it's basically 2N to N, and then all of these 4 are just haploid. Okay. So after the meiosis 1, the cells are already haploid, so they're no longer diploid. And so now looking at meiosis 1, there are the same 4 phases that happen in mitosis. You have prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Okay. Pretty much happens the same way. So in prophase 1, the nuclear envelope breaks down, the centromeres end up, or the centrosomes are on the polar ends of the cell, and your chromosomes condense. Okay. However, one difference in meiosis 1 and mitosis is that the homologous chromosomes pair up. Okay. And so if you remember with mitosis, the homologous chromosomes did not pair up. So let's say you have the paternal copy and the maternal copy, right? They would line up along the metaphase plate like this, right, in mitosis. Okay. That's not how it happens in meiosis. In meiosis, what happens is, instead of lining up singularly like that, the homologous chromosomes will pair up. Okay. So what happens is your paternal and maternal copies will pair together like this, and one copy will be on one side of the cell and the other copy will be on the other side of the cell. So in this case, the paternal copy is on the left side of the metaphase plate, and the maternal copy is on the right side of the metaphase plate. Okay, and so that's very different from lining up like this, right? Because you have, these being split in 2 going to opposite sides in mitosis, but in meiosis, the red one will go to this side and the blue one will go to this side. Okay, and that happens, right? Mitosis is those homologous chromosomes lining up. Anaphase is separating the homologous chromosomes, taking them to the poles, and then telophase is the breakdown of the centrosome, or not the centrosomes, but the mitotic spindle fibers, and then nucleus reforming, and cytokinesis happening. And so let's say, for example, if you have this cell at the beginning, right? So let's say you start with, come on, right? This cell where you have, right, your paternal and your maternal copy. I can get the right color. What your daughter cells look like that you end up with are like this. Right? So one copy, either the paternal or the maternal, will end up in the daughter cell. So now, instead of having two copies of this chromosome, you have one copy. So you're 2N to N. So two copies here, one copy there. Okay. And what's important though is even though the number of copies have been reduced, these chromosomes are still replicated, right? They're still sister chromatids. What also happens during prophase 1, okay, so this only happens during prophase 1, is that the homologous chromosomes will swap pieces of their DNA segments. So this is known as crossing over. So crossing over only happens in prophase 1. Okay, that's the only time crossing over happens. And so the way crossing over happens is, you have your homologous chromosomes pair up, right? Just like this. So you have your paternal and your maternal copy. These chromatids here are coming into contact with one another. And what they can do is they can swap their bits of DNA information, their segments, basically like grafting onto one another. So notice that the paternal chromosome now has maternal segment, and the maternal now has a paternal segment. And so you end up with this area that's crossed over. And so that leads to more genetic variation within the gametes, and leads to more genetic variation in the offspring. Okay. Now, another really important thing that can happen is what's called independent assortment. And independent assortment happens only in metaphase 1. And what independent assortment is, right? So we're going to show kind of one example. Let's say we have a cell with 2 chromosomes. Okay, so you have a big one and a small one. Right, so the big and the small are homologous chromosomes. So they're going to pair up together, line up on the metaphase plate. Right. So what you can do is you could have this situation where, let's say they line up on the metaphase plate like this. So that means that one daughter cell, right, will have a big red and a small red, right, because they're going to go this way. And the other daughter cell will have a big blue and a small blue. Okay. And so you have all the maternal and all the paternal DNA in those daughter cells. Another thing that could happen is, let's say instead they line up like this, where the maternal of the big chromosome is on the right and the maternal of the small chromosome is on the left. So now all of a sudden you have a different scenario of what genes those daughter cells have. So the right side will have the big red and the small blue. The left side will have the small red and the big blue chromosome. Right? So they would look like that, which again is very different genetically from these. Okay. This process of how they line up on the metaphase plate is known as independent assortment. Okay, this only happens This independent assortment only happens in metaphase 1, because after metaphase 1, the homologous chromosomes are split, and you can't, they can't independently assort. Basically, what independent assortment is saying is that if you get the maternal copy of, say, chromosome number 1, that doesn't mean that you're going to get the maternal copy of chromosome number 2. The cell could have the paternal copy of chromosome 2, right? So this could flip-flop. And then another variation would be, say that the big X was on the right-hand side and the small blue was on the left, and then the big red's on the left and the small red is on the right, right? So that's different from this one in terms of what genes the offspring will get from those cells. And so again, that independent assortment is leading to more genetic variation. So two ways that sexual reproduction adds to genetic variation is through crossing over and independent assortment, okay? So those are two big things to make sure you understand. And now this figure here kind of goes through the summary of meiosis 1, right? So remember, you have prophase 1, where the nuclear envelope breaks down. Your homologous chromosomes line up. Crossing over happens, and that's only in prophase 1, right? And then those homologous chromosomes will line up on the metaphase plate. And so notice here that the maternal and the paternal are on different sides, so they've independently assorted from one another. And then when anaphase happens, it splits these homologous chromosomes. So one copy of the homologous chromosomes goes to each pole. Then in telophase 1, the nuclear envelope surrounds it. Cytokinesis happens, and you get two daughter cells. And so remember, these cells are 2N, but by this time they're only 1N. So for example, you have one copy of the big chromosome, one copy of the small, one copy of the big, one copy of the small, but these are still duplicated, right? These are still duplicated chromosomes. Now, these two cells will go into the next division phase, which is meiosis 2. So this cell will divide and this cell will divide. So you end up with 4 daughter cells. And so this is where understanding mitosis really helps, because meiosis 2 is really similar to meiosis. So meiosis 2 involves prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And it happens exactly like mitosis pretty much. So you have your chromosomes, your chromatids, your sister chromatids, and the nuclear envelope breaks down. In prophase 2, those sister chromatids line up along the metaphase plate, right? So now they're lining up singularly, no homologous chromosomes. And then the spindle fibers will pull apart those sister chromatids in anaphase 2. And then you have your sister chromatids going to each pole. And then for each one, you'll have telophase 2, where the nuclear membrane reforms, and cytokinesis happens, where you end up with 4 daughter cells. So notice that each one has one copy of the big, one copy of the small, but the version of those genes are different. So for example, if we look, the big one in this cell is the paternal copy, whereas the little one in this cell is, it looks like a paternal copy that has the maternal crossover on that, and then it's the opposite of these. And then, this one is maternal, and then this one is the maternal with the paternal crossed over. And so none of these cells are genetically identical. Okay, they're very different genetically from the parent cell. Okay, and so again, like I recommended in the video for mitosis, I would go through and draw these two phases, making sure you can understand what happens during each phase. So what happens in prophase 1, metaphase 1, anaphase 1, telophase 1, what happens in prophase 2, metaphase 2, anaphase 2, telophase 2, when you have your homologous chromosomes line up, when interdependent assortment happens, when crossing over happens, when the chromosomes or when the cells go from haploid to diploid. So you want to know all of those specifics that we've talked about. Okay. And so there are some differences in meiosis depending on the gender and what gamete cells that meiosis is making. So for example, males will have 4 viable sperm produced for each cell entering meiosis. So that meiosis cell, divides into 4 viable sperm like we see in this image. However, for females, they have an unequal cytokinesis during meiosis 1 and 2. So 1 egg and 3 polar bodies are produced from every 1 cell entering mitosis. And so only the egg is viable. So you have 1 viable cell from 1 cell that enters mitosis for females, whereas males have 4 viable cells from every 1. And so here is that schematic. So you have your 4 viable sperm cells. Here you have your viable egg, and then those other 3 cells form these 3 polar bodies that exist in that cell. Then you have your sperm to fertilize the egg. You get an embryo, and that nucleus will have 23 plus 23. So you have 23 pairs of chromosomes. So haploid, haploid, diploid. Okay. And so we'll look at an animation kind of going over this, and then we'll finish up talking about how cell reproduction is regulated.
A cell that undergoes mitosis produces daughter cells that are dramatically different from the daughter cells produced by a cell that undergoes meiosis. Let's compare these two processes, beginning with a look at mitosis and meiosis 1. In both mitosis and meiosis 1, cells begin with replicated chromosomes, each cell having 2 chromatids per chromosome. In meiosis 1, the homologous chromosomes pair with each other and come to the middle of the cell together. In mitosis, the homologs move independently of each other. In mitosis, the chromatids separate. In meiosis 1, the homologs separate. In mitosis, a diploid cell divides to produce genetically identical diploid cells. Each cell contains 2 complete sets of chromosomes. In meiosis 1, a diploid cell divides to produce genetically unique haploid cells. Each daughter cell now contains just 1 set of chromosomes. Meiosis 1 is known as a reduction division because the outcome is a reduction of chromosome number. These 2 cells are daughter cells produced from meiosis 1. Let's use just one of them to compare meiosis 2 to mitosis. A cell entering mitosis is diploid, with 2 complete sets of chromosomes. A cell entering meiosis 2 is haploid, with just 1 complete set of chromosomes. In both cells, each chromosome has 2 chromatids. In both mitosis and meiosis 2, the chromosomes migrate to the middle of the cell. Then, the sister chromatids of each chromosome separate to form independent chromosomes. In mitosis, a diploid cell divides to produce genetically identical diploid cells, each with 2 complete sets of chromosomes. In meiosis 2, a haploid cell divides to produce genetically unique haploid cells, each with 1 complete set of chromosomes. If we consider that meiosis 2 actually began with 2 cells, then the result is 4 genetically unique haploid cells. In both mitosis and meiosis 2, the division is not a reduction division. Only in meiosis 1 are the number of chromosome sets divided by half.
All right. And so again, you'll want to make sure you kind of go through mitosis and meiosis and, all of the little details of each division phase and kind of all of the components of those 2 processes. Okay. And so what we're going to finish up now is talking about how that cell reproduction is regulated, and what can happen whenever you don't have those cellular division processes kind of occurring like they're supposed to. For cellular division, an interesting thing about the human body is that not all of the cells divide and reproduce at the same rate. There's this internal control mechanism, and within the cell cycle, there's actually checks and like uh, division checks where it basically serves as like a traffic regulator, traffic cop, in determining when the cell will kind of progress into the next phase and go into the next part of the cell cycle. And so there are checkpoints after the G1 phase, the G going into the G2 phase, going into that division phase, which looks at the cell, checks it, makes sure everything's going okay. And if it's not going good, then the cell will be destroyed, and that kind of prevents kind of bad cells reproducing and prevents things from like cancer and that sort of stuff happening. It's whenever those checks aren't regulating or there's mutations in the proteins and enzymes that, or the proteins that regulate those kind of checkpoints that allow cancer to happen. And we'll talk a lot about that when we talk about chapter 15. There are other outside influences that can modify the cell cycle, so presence of other cells, other growth factors, and hormones. And so we won't go into too much detail other than that, but just know that there are influences outside of kind of these checks that influences the cell cycle itself. Here's a table from your book that kind of shows you cells and how often they divide. So skin cells, epithelial cells, sperm cells will divide constantly, typically throughout your life. After adulthood, cells like your nerve cells, bone cells, and muscle cells stop dividing. And then under certain situations, things like your liver cells will divide if necessary. So like, for example, liver cells don't normally divide in adulthood, but if part of the liver is removed, then they'll divide and replace that. So here's a figure just kind of showing those checks. So you have a check at the end of G phase, going into S phase, end of G2 phase, and then end of mitotic phase. And so checking again to make sure that those cells are like they should be and functioning like they should be before they're allowed to divide and replicate in the body. There are other environmental factors that can influence cell differentiation. So basically what they kind of differentiate or turn into. And so like we mentioned, all body cells, so those somatic cells, right, have the same DNA. But even though they have the same DNA, those cells look and act differently depending on what part of the body they are. So like your skin cells look very different from your nerve cells and function very differently. And so that happens through this process of differentiation, where a cell becomes different from its parent or sister cell, which is ultimately determined by how the genes are expressed in the cell itself. Which is typically regulated by like how often that gene is transcribed and translated into a protein, whether it's present in the cell or not present, whether there's a lot of it being expressed or very little being expressed, so on and so forth. And so after fertilization, the zygote will have a couple of cell divisions where it divides from 16 to 32 cells, and it forms a ball, so what's known as a blastula. After this 8 cell stage, the cells are exposed to basically different environments. So the cells inside of this ball are exposed to kind of a different environment compared to the cells that are outside of the ball. And that determines what those cells differentiate into. So the cells on the outside will most likely be ectoderm, so skin cells, that sort of types of tissue. And then the cells on the inside will most likely be mesoderm or endoderm, which is like your gut lining, your organ tissue, all of those other types of cells. Now, what's interesting is at this 8 cell stage, there's really no kind of differentiation. it's after that 8 cell stage, right? So after the 8 cell stage where they're exposed to different environments. So before this, so 8 cells or less, you can actually stop the division of the cell of the embryo, and you can split all 8 of those cells apart, and you will have 8 identical cells that you then can let divide again, and it can turn into 8 genetically identical organisms. Okay, so this is kind of how cloning, one way cloning can work. So for example, you have your fertilized egg, one division is 2 cells. Those 2 cells divide into 4, and those 4 divide into 8. This is the last point at which all of those cells are exposed to the same environment. Once these cells divide and go to 16 cells, you now have differentiation. You have different types of exposure. And then from 16 to 32, you get even more differentiation in terms of what those cells are exposed to in the environment in which they're growing. And so you start getting ectoderm, endoderm, mesoderm, different tissue layers, differentiation, cells going to turn into cartilage versus nervous tissue, etc. And so again, you can stop it here, pull each of these 8 cells apart, right? So you have 8 individual cells, and then these can be allowed to undergo these division phases and turn into or develop, or develop into 8 genetically identical organisms. Differentiation can also happen later in development. It depends on like when genes are turned on or turned off. And so, for example, webbing between digits are based on whether or not genes are turned on at specific developmental phases. So, for example, ducks and bats have webbing between their digits, and all of that is regulated by a couple of genes called like gremlin, Sonic the Hedgehog gene, and so those turned on and turned off at various times during their development determines whether or not there are webbing between our digits. So if a human has those genes turned on at different times, like they are in ducks and frogs, then they theoretically could develop webbing between the digits. Now, other things that could affect development are external substances that could be harmful. So like smoke, pollution could retard the growth. Alcohol, medications can pass through the placenta. Drugs, the child could be born addicted. Environmental chemicals, so pollution, that sort of stuff, radiation, and then other intrauterine infections like HIV, syphilis, all of those things that they would test a pregnant mother for, just to see if they need to kind of look at the fetus, if whether or not they have those types of complications. And like I had mentioned, you can have reproductive cloning, which you need that undifferentiated cell. So you're actually producing an entire organism, so a copy of that organism during the process of cloning. And so the two methods that you can have, one of those we've talked about, which is that embryo splitting. Another one is somatic cell nuclear transfer. So for embryo splitting, that's where you just split at the 8 cell stage, and you allow those identical cells to grow into genetically identical individuals. So the egg's fertilized, allowed to divide to the stage, you stop it, you separate, and each is implanted into a different surrogate mother, and then it develops. The clones are identical to each other, but they're not identical to the surrogate parents because those aren't the parents that led to the fertilization process, but they would be identical to each other. And then they're also not genetically identical to the parent just because of the way genetics works in terms of sexual reproduction, right? No offspring is genetically identical to their parents. This process hasn't been attempted with humans. It's been done with other organisms. So for example, for like sheep, stop that 8 cell stage, get those 8 identical cells, put them in surrogate mothers, you have 8 identical offspring. The somatic cell nuclear transfer actually produces a clone of the adult. And so the way this happens is you take a somatic cell. So this could be any cell from the body, cell that's not the gamete, because the gamete only has half of the chromosomes needed. Okay, so the somatic cell has those full diploid sets of chromosomes. And what they do is they'll take the somatic cell from the adult that they're going to clone, and they'll insert um, it into a eunucleated fertilized egg. Okay, so basically a fertilized egg um, that has basically had the nucleus removed. Electrical current is used to fuse the cells together, and then that cell is implanted into a surrogate and allowed to develop. And so the offspring would be an exact clone of the organism, the adult organism that the somatic cell came from. So for example, here you have an individual where a somatic cell is taken. You remove the nucleus from that. I'm sorry, you have an egg. You remove the nucleus from that egg. Then you take the somatic cell, the DNA from that somatic cell, and put it into that unfertilized egg that the nucleus was removed from. Okay. Then you put the electrical current, which fuses those cells together, and then now you insert that into a surrogate mother, and you get identical clone of the individual, which would be this one, that provided the somatic cell. Okay, so just again for clarification, because I was, can't talk tonight. You have an unfertilized egg, so an egg cell, remove the nucleus from it. Then you take a somatic cell from the individual you're copying, put it into that unfertilized egg, apply electrical current, fuses the cells. You take that fused cell, insert it into a surrogate, and then the identical offspring will be born. So one final thing is that there are therapeutic cloning mechanisms that have been looked at in terms of like cloning tissues and organs, where we could potentially use this to treat patients. So for example, you could remove a single cell from a patient, use it to, say, grow a heart for a patient that needs a heart transplant, and it would match because it was grown from their cells. And so you could use this theoretically for like creating new cells, new tissues, new organs. So this is just kind of theoretical, not really realized yet, but if it does become realized, it could be a really big thing, especially because we have, issues with donors, organ shortages, all of that stuff. So would be a pretty cool thing to see exist and see happen. Okay, and so that's it for our chapter 17 discussion. That's it for the module. So as always, let me know if you have any questions, any concerns, and we will see you in the next one.